taxonomical classification. We are following Whitaker's system of classification. The 5 kingdom classification, already we have studied about kingdom monera, which is the first kingdom, kingdom protista the second one, kingdom fungi, kingdom plantae and kingdom animalia. Now in kingdom monera we have studied about bacteria, archaebacteria, cyanobacteria and mycoplasma. So kingdom monera are prokaryotes, whereas the remaining 4 kingdoms are eukaryotes, and we have already studied about the differences between the prokaryotes and eukaryotes. Now in kingdom protista, which is the second kingdom of Whitaker's classification, we are following Whitaker's system of classification, whereas living organisms are classified into 5 kingdoms. Then protista, which is the second kingdom, includes eukaryotes, and these are unicellular eukaryotes, whereas monera are unicellular prokaryotes. And most of these organisms under this, they are planktons, means floating, and nutrition is both heterotrophic and as well as parasitic. So some are parasites and some are saprophytes, heterotrophic nutrition. Then when we come to the reproduction, so reproduction is by sexual and asexual type of reproduction. Then some of them are having cell wall, and certain organisms, they do not have cell wall, they are having a thick layer, a soft layer which is called as pellicle. So then protista are further, kingdom protista is further divided into 5 divisions. So the first division, Chrysophytes are the first division. Then Dinoflagellates, second one. Euglenoids, third one. Then slime molds, fourth group. Then Protozoa is the 5th division. So there are 5 groups under kingdom protista. So Chrysophytes, Dinoflagellates, Euglenoids, slime molds and Protozoa. So protista are unicellular organisms. Monera are also unicellular, but monera are prokaryotes, protista are eukaryotes. Then coming to the characters of this each. So we are not studying the details of each division. So 1 or 2 characters and examples, one example. So Chrysophytes, they are unicellular organisms. They are found in fresh water and as well as marine. We come across both type of organisms. Now in this we come across examples like diatoms and desmids are examples. So Dinoflagellates are also unicellular. Now one by one, Chrysophytes. So Chrysophytes, they are also commonly called as golden yellow algae in generally. So they are also one type of algae. Under this we come across diatoms. Now about diatoms, we are studying about one more part there. So diatoms are peculiar organisms. Though they are unicellular, they are somewhat peculiar when we compare to other unicellular organisms. They are made up of cell wall, The cell wall is made up of silica and pectin, and these diatoms, the cell wall is made up of two parts. So this diatoms, for example, if you take the cell structure just to give an idea, so this is how the cell looks like. The cell wall is not entire. Now whenever we draw a cell, so we draw continuously like this. That means the cell wall is continuous in a plant cell or like that. But in the diatoms, the cell wall is made up of two parts, or in other words, two pieces, such that one part is fitted into the other. So the two halves, each half, so one of the cell wall is somewhat larger, the other one is slightly smaller. So it is fitted into each other, just like a soapbox. So exactly. So we are referring a soapbox because in a soapbox usually the lid and the container will almost are of same size. But when we say about a tiffin box or something like that, the container will be larger and the lid will be smaller. But here the two cell walls are of equal size, and that is why these are somewhat peculiar. So, but however, as these are found floating on the water, so these two parts which are fitted into each other, they do not separate easily. They are exactly or tightly fitted into each other. Now that is a peculiar nature of these diatoms. Now next thing is, this cell wall is made up of silica and pectin and certain other compounds which are of minute proportions. So mainly silica and pectin are the chemical composition of this cell wall. Now after the diatoms, they undergo death. When that cell completes its lifespan, it undergoes death. These cell walls, that cell becomes empty, and the cell wall remains because it is a non-living thing. So this cell wall which is present, it settles down in the water. So when it settles down, suppose when they are present in oceans, they settle down at the bottom of the ocean. Suppose when they are present in the lakes, it settles down at the base of the floor of the lake. And this cell wall goes on deposit. So over many years, say about million years or billion years, the cell walls which are deposited, so these that dead cell wall, the dead cells, the remaining cell walls of the dead ones, they get deposited and they form what is called as diatomaceous earth. So this diatomaceous earth, it is formed at the floor of the ocean or floor of the lake or any aquatic ecosystem. Now this diatomaceous earth is useful for various purposes. Now it is extracted, it is removed from that region, and it is used as for filtration of oils and for manufacturing polishes. So polishes means this metal polish and as well as even shoe polish. So any material which is to be polished with that material, so that is used in the manufacture of that purposes. So diatomaceous earth is used for manufacture or preparation of polishes, filtration of oils, then even purifying water, and it is used as an insecticide, and it is also used for the increases as a treatment for the joints in case of bones and all that. So it is used for various purposes. In addition to this, diatomaceous earth is also used to increase the magnifying power of the lens, especially in camera and also in the specs, where the magnifying power is to be increased or it should be properly where it is to be tested there. So there it is used for that purposes. That is one use. Then it is used as a soundproof material. There when the walls are to be constructed, when it is mixed with the diatomaceous earth, that wall or that part of the building or that construction, it becomes soundproof. Means it does not allow any sound waves to be passed through it and to be heard on the other side of the construction building. That is the use of this diatomaceous earth. So the uses of diatomaceous earth are still many more, and even in the road paints there also it is used, because road paints are somewhat specific, and that is why this diatomaceous earth is used. Then the Dinoflagellates, these are another group of organisms coming under kingdom Protista. Di means two actually. Flagellates, flagella, they possess two flagella. Of course these two flagella are of unequal size. That is the main character of this group. So the two flagella are of unequal size, and of course they help in the locomotion. These dinoflagellates are also planktons. They are found floating on the sea or like that, and then when they are in large proportions, under this we come across this Euglenoids is one example. So Euglenoids, and many more examples we come across under this. So dinoflagellates, when they are present in large populations, especially in oceans or as a planktons, they appear red in color. Means they form what are called as the red tides. So when that water appears red, this water appears as a red color, so there are red tides. So dinoflagellates cause what are called as red tides. That is the characteristics of dinoflagellates. Then Euglenoids, these are also having two flagella, just like dinoflagellates. So of course under this Euglena is the example. So Euglena is one. Under dinoflagellates, Noctiluca is also another example. So Euglenoids, they are one group of organisms which are having two flagella, but they are having no cell wall. Cell wall is absent in this organism, Euglenoids. And Euglenoids, they instead of having a cell wall, they are having a protective layer called as pellicle, and that is a characteristic feature of this Euglenoids. Though they are having two flagella, one flagella is very small and one flagella is very long. But here these in dinoflagellates, they are almost lengthy, but they are unequal in size. That is the difference between the Euglenoids and dinoflagellates. But Euglenoids, they do not form red tides. But dinoflagellates, when they are in large populations, that water appear as red in color. So when the waves are moving along that, so there appears red tides, that water appears red color. That is about Euglenoids, which are not having any cell wall, and they are having a thick layer called as pellicle. Then slime molds. Slime molds are also unicellular but multinucleate organisms. So each cell contains many nuclei, and these slime molds, they are usually saprophytic in nature. They are found in decaying leaves, decaying woods or like that, and even in wet areas also they are found. These slime molds, They are unicellular and they are having many nuclei. So whenever they reproduce, they aggregate together to form this multicellular cellulated nucleated structures, which they form what are called as plasmodium. So plasmodium is the name given to the aggregated structure of the slime molds when they are presented together and involved in reproduction. That is the slime molds. In this, for example, we can take Stemonitis is one example. This is one example of slime molds. Now the fifth group of the kingdom Protista, which is also unicellular. However, Protozoa under Protista, Protozoa is the fifth group. However, in the other classifications, certain classification systems, they classify Protozoa under kingdom Animalia. So this Protozoa, when we study under kingdom Animalia, we come across phylum Porifera, Coelenterata, Platyhelminthes and so on. So actually, according to some, Protozoa is the phylum which is considered as a phylum and included under kingdom Animalia. But according to Whitaker's classification, which we are following, Protozoa group is included under kingdom Protista. So this Protozoa, which is unicellular, it includes 4 group of individuals again. So there are 4 type of amoeboid protozoa, flagellated protozoa, then ciliated protozoa and sporozoa. So these are the 4 groups of protozoa. So depending upon the locomotory organ which is present, they are classified into 4 groups. So what locomotory organ they possess? So in case of amoeboid, so the word itself suggests amoeba is the example in that. So here pseudopodia is the locomotory organ. So though they are unicellular, so the amoeboid protozoa, they possess a pseudopodia as the locomotory organ, and of course there is the contractile vacuole which helps in the osmoregulation of that cell. So that is amoeboid protozoa, where the pseudopodia helps in the locomotion. Now flagella are the locomotory organs. So in this amoeba, ant amoeba are the examples. So amoeba is a freshwater organism, whereas ant amoeba is a parasite which cause amoebiasis. Then flagellated protozoans. So these flagellated protozoans, for example, Trypanosoma is one example. Trypanosoma, it is a parasite actually. It possesses a long flagella and which helps in the locomotion. So Trypanosoma causes sleeping sickness. So there are different types of species under this Trypanosoma which cause different types of sleeping sickness in the human beings. Then ciliated, in this we come across Paramecium. So Paramecium is the one where the cilia which are present all around the cell, which are small hair-like structures, but flagella is a long thread-like structure which helps in locomotion. Cilia are very small. They are small hair-like structures which are surrounding the cell, and they help in the locomotion. Now another characteristics of this ciliated protozoa is there is a small opening at one side of the cell, and this small opening, which is generally called as the mouth, it leads into a canal which is called as a gullet, through which the food enters and is obtained by the organism. That is ciliated protozoans. Now sporozoa, they don't have any locomotory organs. That means there is no pseudopodia, no cilia, or even no flagella. And under this we come across the example Plasmodium. So Plasmodium is an example which causes malaria. So Plasmodium is again of different species, Plasmodium falciparum, Plasmodium vivax, and so on. All these cause malaria. So they don't have any locomotory organs, and they occur as small structures like spores, or which are called as sporozoids. And those are the 4 types of protozoans based on the, or classified on the basis of this locomotory organs: amoeboid, flagellated, ciliated, and sporozoa. So that is about kingdom protista. Then coming to the next kingdom, the third kingdom, kingdom fungi. So kingdom fungi is also called as kingdom mycota. So kingdom mycota, which is the third kingdom, it is also a eukaryote, but here we come across multicellular organisms. So unicellular we come across only in protista and monera, but however there are some unicellular in fungi also. When we study the different groups, we come across those examples. Now fungi or this mycota organisms, they don't have chlorophyll at all. So that is why basically they are achlorophyllous organisms. Organisms without chlorophyll. So they never possess chlorophyll at all. That is the important character of this fungi. So when they are not having any chlorophyll, definitely they have to depend on nutrition on an external source. Just as in bacteria we have studied autotrophic and heterotrophic. So autotrophic means they prepare their own food material either by photosynthesis or by chemical oxidation. But in heterotrophic, the nutrition they have to depend on the external source itself. Same way here, these are heterotrophic, so they are either saprophytic or they are parasitic. So they never contain chlorophyll at all. Then if you come to the structure of the fungi, any fungi, basically the body of the fungi is made up of thread-like structures called as hyphae. So hyphae are minute thread-like structures which are present in the body of the fungi. Fungus single, like that bacterium single, bacteria plural. So in any fungus you take, the body of a single fungus, there the body consists of fine thread-like structures which are called as hyphae. Now these hyphae, they may be branched or unbranched. Like that, the hyphae form a interconnected network. So they completely form a network, interwoven network. So that interwoven network is called as mycelium. So hyphae and mycelium means they are related. Hyphae is the single thread-like structure, but this hyphae, when it is present in large numbers, either branched or unbranched, they form a mass called as mycelium. So mycelium is the body of the fungus. Then if you come to the reproduction, so in the case of this, we find both asexual and sexual type of reproduction in this. In case of sexual reproduction, there are again 3 types of methods taking place in the fungi: plasmogamy, karyogamy, and meiosis. So this meiosis occurs after the formation of the zygote. So in plasmogamy, just we are not studying about the detailed methods about that. So cytoplasm, we say, it is actually whenever the cells of the fungus, they fuse each other, only the cytoplasm fuse, but not the nuclei. The nuclei remain separately, and that is plasmogamy. But in karyogamy, what happens? Nuclei fuses, the nuclei of the two cells. Karyo means actually nucleus. So these nuclei will fuse and further involve in reproduction. So how that further development takes place, we are not supposed to study in detail. And again, when the gametes are formed, so usually in case of sexual reproduction, the male and female gametes which are formed, they involve in the formation of zygote. So that zygote undergoes meiosis and further develops to produce spores. This is how the sexual reproduction takes place in the fungi. So mainly, whenever we remember about fungi, we should remember that they are achlorophyllous organisms, one, and they are consisting of hyphae. The body is made up of thread-like structures called hyphae. The mass of hyphae, which is interwoven, and that forms the mycelium. And then, of course, nutrition is heterotrophic. It is either saprophytic or parasitic, because there is no chlorophyll at all. And the sexual reproduction takes place by plasmogamy or karyogamy, or either by meiosis. That is about the group characters of the group or the kingdom fungi or mycota. Again, when we come to the classification of this, as protista are divided into 5 groups and protozoa again into 4 groups. Fungi also is again divided into 4 groups. So those 4 groups are: Phycomycetes is one group, then Ascomycetes, then Basidiomycetes is another group, then Deuteromycetes. So these are the 4 groups of, or the divisions of kingdom fungi or kingdom mycota. Phycomycetes, these are again classified on the basis of the reproduction or the nature of the hyphae. Mainly reproduction is considered here. In case of Phycomycetes, the reproduction takes place by structures what are called as zoospores or sometimes planospores. These are the reproductive structures under Phycomycetes. Then actually myco means actually fungi, phyco means algae. So these organisms, fungi, they actually resemble algae in some characters. That is why they are called as Phycomycetes. Mycetes means fungi, myco, fungi. So phyco means algae. Fungi resembling algae in some characters. Means when we study the details of all those characters, we see that they almost resemble in the algae, because we have come across zoospores in algae also, and that is why they are included. And about hyphae, one point to just mention here before going to this. Now hyphae means they are small thread-like structures like this. So these thread-like structures, when they are observed under the microscope, they are made up of cells like this. Suppose this is one hyphae. So the hyphae is made up of cells. This is one hyphae like this. So this hyphae consists of elongated cells which are having a common wall in between. Suppose this is a cell, so this is the common wall in between. So this cells are in contact with the cell walls like this. That is how the hyphae is made up of. Hyphae is made up of many cells. That is why fungi are multicellular. They are multicellular or eukaryotes. Suppose this is an hyphae which is made up of cells which are arranged in a row. Now this hyphae sometimes when the cells are arranged with the cell walls attached with each other, it is called as septate hyphae. That means in between two cells there is a partition, there is a wall which separates them. Septa means wall, partition. So when the partition is present in between two cells, it is septate hyphae. Sometimes in certain hyphae the partition is absent. Suppose if there is no partition between the two cells. Of course this is one cell and this is another cell, but the cell is not having any common wall in between, or in other words there is no septa, there is no partition wall in between the two cells. So if this is the case, it means that the cytoplasm of the two cells are in directly contact with each other. So there is continuation of the cytoplasm of the two cells. That means there is no septa. So such hyphae is called as aseptate hyphae. So when we study about the characters of fungi, of course fungi, the body of any fungus is made up of hyphae, thread-like structures. But again there are two types of hyphae. The hyphae may be either septate or aseptate. If there is partition between the cells, septate. If there is no partition, aseptate. That is how septate and aseptate hyphae can be distinguished. So here in the four groups what we are studying, in case of Phycomycetes, we come across aseptate hyphae. Aseptate hyphae is found in the Phycomycetes. That means this type. In the remaining we find septate. That is how we have to remember the characteristics of these groups. Phycomycetes are aseptate. That means they resemble the algae, hence so the cytoplasm are continuous and the hyphae appears like a tube-like structure where the cytoplasm is present and the nuclei are freely suspended in the cytoplasm. They are present freely in the cytoplasm. That is the aseptate. So we sometimes when we observe this aseptate, we believe that this is a single cell and this cell contains many nuclei. So when many nuclei are found like this freely present, sometimes the word cenocytic is also referred. But however, we are using commonly aseptate hyphae, wall is present, absent. Septate, wall present, aseptate, wall absent. So Phycomycetes are aseptate. And these they reproduce by the help of zoospores. Then examples under this we come across Rhizopus and this Phytophthora. So Rhizopus, which is commonly called as a mold, or it is also called as bread mold. So bread mold, it is a common example which everyone has seen this in our home. So in your homes or in your house, especially in the kitchen, we come across this mold. So mold in Kannada, boost it is called as. So whenever you keep a bread or any eatable like roti for a long time without observation, So you have kept it without noticing that. You can see some cotton-like mass appearing on the surface of that eatable, either bread or this roti or even any other material. Even on fruits, if you cut a fruit, apple or like that, and you keep it unnoticed for a long time in a particular place, so you can see the growth of the fungus on that surface. So that is nothing but bread mold, what we call it as boost. Now kannadadolaga boost anta ನಾವು ಕರಿತೀವಿ, ರೊಟ್ಟಿ ಬೂಸ್ಟ್ ಬಂದ್ಬಿಟ್ಟವು ಅಂತ ಹೇಳ್ತಿರ್ತೀವಿ. That is actually rhizopus. So rhizopus, actually there are different types of molds. Mucor is another type of mold, and again when we go into detail about that, the color of the mold may be different. Sometimes you can see black color mold in some fruits or eatables. Sometimes you see some gray color or somewhat light yellowish in color, so somewhat white color. So different molds are there. So it is an example of Phycomycetes. So when you closely observe that mold on that eatable, either fruit or bread or like that, so when you closely observe keenly, you can see small thread like structures on that surface. So ನೀವು ಆ ರೊಟ್ಟಿ ಅಥವಾ ಬ್ರೆಡ್ ಮೇಲೆ ನೋಡಿದ್ರಿ ಅಂದರೆ ಸಣ್ಣ ಸಣ್ಣ ದಾರದಂಥ ರಚನೆಗಳು ಬರ್ತಾವೆ. ಸೊ ದ್ಯಾಟ್ ಈಸ್ ನಥಿಂಗ್ ಬಟ್ hyphae. So when you take a single thread or a single fiber like structure and observe under the microscope, you can observe this type. That means in case of this, it is a septate. In this other, there are septate hyphae. That is how this aseptate hyphae is found in the Phycomycetes, and thus Rhizopus and Mucor, they are actually called as molds commonly. Mold is the common name. Rhizopus is one scientific name according to binomial nomenclature. So Rhizopus is the genus name. Albicans, actually it is the species name. So we are not studying the complete scientific name of the organism. Just we are mentioning the genus name or the common name of this example as an example of Phycomycetes. Then coming to the next group, Ascomycetes. So the reproduction as the zoospores, here there are what are called as ascospores. That is why Ascomycetes. Now these ascospores are produced in structures called as ascus, single ascus, plural asci. So usually when there is a word us for single, I is the plural for that. For example, when you take nucleus, when you study about the cell, nucleus single it is. When you say many, when there are many nucleus like that, nuclei is to be used. The plural for nucleus, nuclei. Same, ascus single. So ascus is a small structure which is formed in this, and there we find the structures called as ascospores in that bag-like structure. Actually, ascus is word is a Greek word which means bag-like structure. So that ascus is a bag-like structure which produces the ascospores, and those ascospores help in the reproduction. And in this there are again different examples, and one example is Penicillium is one example. So Penicillium is one, and there is one more called as Aspergillus, which are made up of hyphae. So these three, they are septate and branched hyphae. See, in Phycomycetes, a septate, but in the others we come across a septate hyphae like this. So penicillin is also made up of. Penicillium means it is a fungus which produces penicillin antibiotic. We have heard about penicillin injections which are given. So penicillin is a antibiotic, which is a chemical which is obtained from Penicillium fungus, and Aspergillus is another fungus. So one more important thing about Ascomycetes is there is one example called as yeast, which is scientifically called as Saccharomyces. Yeast is a common word. Saccharomyces genus, Saccharomyces cerevisiae. Cerevisiae means it is the species name. So Saccharomyces is used for yeast, and this is the only example for unicellular fungi. It is an exceptional one. But however, when we say that fungi are made up of hyphae, they are multicellular. It is the general character with one exception. So in kingdom fungi, there is only one exception that we should remember. The only unicellular fungus is yeast or Saccharomyces. So yeast is made up of small cells like this, but not hyphae. There is no hyphae in yeast. The yeast which we commonly use in the preparing bread in bakeries, even in preparing in idli and dosa, there also it is used. So that is the yeast which is used. It is the economic importance of yeast. Now, why this yeast is used in that bread making or cake making cakes and all that? So yeast contain a particular enzyme called as the zymase actually. So the cell which is present, that is a single cell of yeast, contain an enzyme called as zymase. So this zymase helps in the softening of that material. So that is why the bread, cakes and even idli and all that, so they become soft because of the effect of the enzymes. So this is the example of group or the division Ascomycetes. So the ascospores, septate hyphae, ascus structures which involve in the reproduction, and they are the examples of the Ascomycetes. Then Basidiomycetes. Here again septate hyphae is same, common, but reproduction takes place by structures called as Basidiospores. That is why Basidiomycetes. So ascospores, ascus, they are present. Basidiospores, they are present in Basidia. So like that. So Basidia they are the structures which contain Basidiospores. They involve in reproduction. They are septate, and these Basidia are present in specific structures called as this Basidium, or what are called as gills and all that. So when we study about the details of the reproduction, so Basidiospores, Basidia are present, ascospores, and here zoospores, which are similar to algae, and they are septate and branched hyphae. Then when we come to this another important character, they are usually, the mycelium is dikaryotic. Dikaryotic. That means usually, I said septate means this one. Usually when we say a cell, there is a single nucleus. This is the nucleus in each cell. But when in case of Basidiomycetes, the cell contains the two nuclei. That is why dikaryotic. Di means two, karyo means nucleus. So just I mentioned here, plasmogamy, karyogamy, meiosis. Plasmogamy, it is a fusion of cytoplasm, fusion of nuclei. Karyo means nuclei, remember. Whenever you come across a word karyo starting, or the karyo word is there in that, karyo part is there in that word, it means that it is concerned with nucleus. So here fusion of nuclei, and here presence of nuclei. Di means two. So two nuclei are present in the hyphae or the mycelium of this group or this examples. So Basidium is the main important character. And again when we come across this, so Agaricus is the example. That means commonly what we call it as mushroom. So which is edible. Nai kode anabe anta navu karitivi ade mushrooms, Agaricus. So they are actually rich in protein. So mushrooms are edible, but however mushrooms, the certain mushrooms are also poisonous. All mushrooms are not edible, remember. So under mushroom is a common word. Agaricus is the genus name. So there are different genera for this. So mushrooms, they are rich, or they are utilized for the proteins. They are edible because of their rich proteins present in them. So this is one example. And even another example is Puccinia. So this Agaricus and Puccinia and Astilago is another example. So where this is commonly what we have seen. So under each group already we have seen one example. Rhizopus everybody has seen in their homes. Yeast, it is commonly used in making bread, cakes, etc. And Agaricus, we see an umbrella-like structure. What we see, it is nothing but the Agaricus. So this umbrella-like structure which is seen, it is the Agaricus. It is edible because it is rich in proteins. And in this body of the Agaricus we find hyphae. So though it is umbrella-like, when we analyze that, when we observe the structure of that, actually that umbrella-shaped structure is called as pileus. So that part is called as pileus. It contains basidia. So inside this, when we observe the umbrella-like structure, inside that we find flat structures which are radially arranged inside. So how the this rods, small metal rods are arranged in a umbrella inside. So like that we can see plate-like structures arranged radially inside the pileus of that umbrella-shaped structure. So those plate-like structures are actually called as gills. Gills, the word is also used in fish. That there it is respiratory organs. But here the same word is used. So gills are structure which are present in this Agaricus. In each gill there are basidia. And each basidia, basidium single, basidia is a plural. So each basidium contains basidiospores. So like that the reproduction continues. So whenever the Agaricus has to reproduce, imagine so that umbrella shaped structure which is having the basidia inside, there the basidiospores which are produced there, they fall downwards. So these basidiospores will fall, and these basidiospores they are carried by wind or water to different places. When these basidiospores, which are unicellular structures, when they fall in a suitable medium like decaying leaves or any organic matter, decaying organic matter, when they fall there, that basidiospore will germinate and produce the hyphae and the mycelium. That is how the reproduction takes place. Even in the case of these ascospores, zoospores. The zoospores which are produced by Phycomycetes. For example, if you take the mold, to give an idea of the spores, suppose when we speak of this Rhizopus mold, we believe that mold is present only in the kitchen part of our house. So that means we see this mold in the fruit, any eatable items, isn't it? But even if you keep this item like bread or roti like that away from kitchen, there is the appearance of the Rhizopus. So even if you keep a piece of bread in our classroom where there is no kitchen at all, in our school, so the school is away from the house, isn't it? So even if you keep a piece of bread or roti or like that in the classroom, after some days you will see the appearance of the Rhizopus. Now the question is, from where this Rhizopus have appeared, or how this Rhizopus have been developed? It is because of the spores. So spores are spread everywhere. Even spores are present in front of us. So these spores, when they come in contact with these such eatables, they develop into mycelium, and they grow on that part of the food material. That is how Rhizopus reproduces, or even any fungus reproduces by the help of spores. Either it may be zoospores, aplanospores, or it may be ascospores, or it may be basidiospores. So all these spores are distributed. So one agaricus, if you see anywhere, which is umbrella, it is producing millions of spores, not thousands. So there are millions of spores, and they are distributed in all directions, but all the spores will not develop into a new organism, because most of the spores, or some of the spores, they reach a unfavorable place which is not suitable for development. So they may fall on a dry place, they may fall in water, and they get destroyed. But those which which are carried by wind or water to a favorable medium or a suitable medium, so there they germinate and grow into hyphae and mycelium. That is how these basidiospores, we see about the growth of this agaricus. So whenever in front of your house or somewhere under a tree, we may see an agaricus. So nobody has come and grown agaricus in that area. By naturally it has grown there. The basidiospores have been carried from somewhere else. So there was from somewhere else means where agaricus has been grown. It may be far away from this place. So it has been, the basidiospores are carried by wind. They are carried by water, either rainwater or like that. So these basidiospores, when they fall on a suitable medium, they germinate and grow into that new organism. That is how this agaricus or whatever it is, all these are developing. Now in Deuteromycetes, these are some different group of fungi, and that is why they are called as imperfect fungi. So these imperfect fungi, they are mostly saprophytes, whereas some are parasites. And here also there is septate hyphae. And another important character is we find only asexual reproduction in this group. But in these three we find both. So even in these we find sexual reproduction. which I have already mentioned here. It may be either plasmogamy, sexual reproduction takes place either by plasmogamy or karyogamy or even by meiosis. So meiosis means it is a meiosis of the zygote. The gametes which are formed, when they fuse to form the zygote, they undergo meiosis. That is one type of sexual reproduction. Fusion of the cytoplasm, plasmogamy. Fusion of the nuclei, karyogamy. But in Deuteromycetes, we don't find sexual reproduction at all. And because of that, imperfect fungi. That means this character is not present in this group. So they are imperfect. And in this, we do not come across a common example, but these are some rare examples which we cannot see commonly. But however, in the first three groups, at least one example we have seen them. Rhizopus we have seen, yeast and agaricus. But Deuteromycetes are one group where the organisms are not having complete characters of the fungi. Only asexual reproductions takes place here, and it is septate hyphae, of course. And there are certain examples like Coletotroicum, then many others like Alternaria is another example coming under this group. So these are the four groups of kingdom Mycota or the kingdom fungi. Then after studying these three kingdoms, we come to the next kingdom actually, but that next kingdom is studied as a separate chapter. That is the third chapter, which is kingdom Plantae. But after this, there is one small aspect which we have to study. That is about virus. So virus, which we have already heard about this. So it is so virus is considered as an organism. So presently we are aware of the COVID-19. It is a virus. So AIDS virus, HIV. Now this virus has not been included under any kingdom according to Whitaker. So Whitaker's classification is about living organisms, monera, protista, then fungi, plants, animals. Now virus has not been included, but however we are studying this under this chapter. Now whether virus has been included under any kingdom or not, we are not bothered about that. But we are bothered about the characters of the virus, what this virus is made up of, and about that. But however, not in detail. Now this virus, of course, according to other kingdom, for example, when we study about the classification, Copeland, who classified organisms into three kingdoms, then Ernst Haeckel, when he classified living organisms into two kingdoms, and again one more classification, which is living organisms are classified into 6 kingdoms, 7 kingdoms. There we find viruses are included under one of the kingdoms. But here, according to Whitaker, viruses are separately studied. They are not included under any kingdom. So when we are studying in this chapter, means viruses are not included under either protista or fungi, but they are separately to be studied. So virus, briefly, viruses were first discovered by a Russian scientist called as Ivanovsky in 1892. And when he was studying about tobacco disease, Up till 1892, all diseases which were seen in plants and animals were thought to be by bacteria. Any disease which is observed in plants or animals, it was believed it is caused by bacteria. But when Ivanovsky, he was studying a disease called as the tobacco mosaic disease, means in tobacco plant he was studying about mosaic disease, and initially he thought that it is due to bacteria. So but when he studied about that disease, he found that this tobacco mosaic disease is not by bacteria, but it is due to some other organism which is smaller than bacteria. See, bacteria are unicellular, which we have studied in kingdom monera. Either it may be coccus, bacillus, or whatever it is. So bacteria are unicellular, but these viruses, Ivanovsky came to know that they are smaller than bacteria, and these cause mosaic disease, but not bacteria. So that is how viruses were discovered, and later various other scientists studied about viruses. But however, Ivanovsky did not studied more about virus, and he did not explain the structure of the virus even. And then Wendell Stanley, another scientist, he in the year 1924, he studied about virus and found that viruses have a particular structure, and viruses are not made up of cells. See, the viruses actually they are not made up of cells. Then viruses, even if they are not made up of cells, so they cause disease. They express living characters, and viruses can be seen only under electron microscope. They cannot be observed under compound microscope. So the compound microscope means the microscope which we commonly see in our high school laboratories and the college laboratories. They are all compound microscopes. But viruses can be seen only under electron microscope. Then viruses, they possess both living and non-living characters. So when they are not made up of cells, what they are made up of? They are made up of nucleic acid and a protein cover. Any virus you take, it contains nucleic acid, either DNA or RNA. Both are not present together. If you take any virus, it contains RNA or DNA. So in the COVID-19, we find RNA virus, but no DNA. So but in any other virus you take it, there may be only DNA. So a virus contains either one of the nucleic acid, DNA or RNA, and that is surrounded by a protein layer. There may be some carbohydrate molecules or fat molecules, but the main component is protein. So that is how a virus is made up of a protein coat. So that protein coat is actually called as capsid, that outer part. Inside there is the DNA or RNA. Then viruses, they possess both living and non-living characters. So whenever viruses, they express living characters when they enter into the cell. When they enter inside the cell, they express living characters, they reproduce. So when they are outside the cell, they remain inert, and they appear as a non-living thing. That is the non-living character of virus. And viruses can be stored in bottles like chemicals. So that is non-living character of virus. And the viruses are not made up of cells. That is also a non-living character. Viruses contain nucleic acid. So wherever nucleic acid is present, it is the living character. So either DNA or RNA, where it is present, it expresses the living character, and that is about this virus. So that is the living character. So they reproduce inside living cells. So already we have mentioned growth and reproduction are the main characters of living. So here also viruses reproduces only inside living cells. Hence that is the living character of the virus. And viruses, they produce various diseases. as we mentioned in bacteria like cholera, tetanus and like that, viruses produce other diseases like polio and this mumps, common cold and this HIV, AIDS, all these are the examples of this viruses. So viruses, they are producing diseases and they cause diseases when they infect the cells, when they enter into the cells. That is in brief about a virus. And then there is one more aspect called as viroids and lichens. Just the meaning of this, what are viroids and what are lichens? Viroids are certain RNA molecules which are causing infections, infectious RNA molecules. So when we say DNA and RNA, actually they are useful in our cells in generally. DNA is the main component of nucleus. It is very useful. RNA is actually helpful in protein synthesis. But exceptionally, there are some RNA molecules which cause the disease, which are infectious. So such RNA molecules are called as viroids. So what are viroids? So viroids and viruses are different. Virus is separate. It is made up of protein coat and nucleic acid. That's all. No cytoplasm, no nucleus, no cell wall or something like that. No cell at all. But viroids, they are RNA molecules which are infectious. Now this RNA molecules, when you say they are infectious, they cause disease. So they also cause the disease in plants and animals. But so far there is one example in a potato plant which causes called a potato spindle disease. So where actually the potato tubers which are formed in the plant, they are affected and they become spindle shaped. So when we say potato, we usually see that potatoes are somewhat like this, isn't it? But here actually what happens, the potato becomes small and flat and elongated. So they become pointed at both ends and become very narrow, very small in size. So that is potato spindle disease, which is caused by viroids. So what are viroids means? Infectious RNA molecules, or in other words, RNA molecules which cause the disease are called as viroids. Example, potato spindle disease. And hepatitis, when we say in case of human being, there are different types of hepatitis. Hepatitis A, hepatitis B, hepatitis C, and now it has been found that hepatitis D has been caused by viroids. So whatever it is, what are viroids? Example. That is enough about viroids. Then lastly about lichens. Lichens are organisms which are of symbiotic associations. Symbiotic means close association between two types of organisms. So a body of a lichen is made up of fungi and algae. So lichen is an organism which contains fungi as well as algae. So here algae conducts photosynthesis. Fungi helps in absorption. So when there is mutual help between two organisms, it is called a symbiotic relationship. When I am helping a person, he is in turn he is helping me, means it is symbiotic relationship. So that means mutual help. So lichen is such kind of organism which contain fungi and algae. Fungi is helping algae, algae is helping fungi. So fungi absorbs the water and minerals and provides to algae, where algae conducts photosynthesis and provides food to the fungi. So the part of the fungi in the lichen's body is actually called as mycobiont, and this is called as phycobiont. So mycobiont means the fungal part, the part of the fungi in lichen, that's all. What is mycobiont means the part of the fungi in lichen, and what is phycobiont means the part of the algae in the body of the lichens. Then lichens, about lichens, this is one example of lichen which is called as stone flower in commonly. Now lichen is also used as an edible component. Even we also use lichens unknowingly without our notice. How? So lichens, one lichen which is called as stone flower, kalhu anta ನಾವು ಕರಿತೇವೆ ಕನ್ನಡದಾಗ. Now when you go to the market and purchase masala, means not powder, so different items are kept in small boxes when you go to the market there. So different items are taken, leaf like structures, small stem like structures, they are collected. In that one of the component is lichen, which is somewhat folded like and black and white in color. ಅದು ಒಂಥರ ಹಾಳಿ ಇದ್ದಂಗೆ ಇರ್ತದೆ ನೋಡ್ರಿ, ಕರ್ರಗ ಮತ್ತು ಬೆಳಗ್ಗೆ ಇರ್ತದೆ. So that is lichen which contains some ingredients for that masala. So that is where lichens are economically used. Now lichens, another important thing about lichens. So this is one example. So what are lichens? Lichens are organisms which are having a symbiotic association of fungi and algae. The fungal part is called as mycobiont, the algae part is phycobiont. Algae is phycobiont, fungi is mycobiont. Example, this is actually examples are not helpful for us, means it is not asked. Another last point about lichens is lichens are the indicators of pollution. How? Now whenever in a area, suppose there may be a forest area or there may be some field or like that, if there are lichens growing in that area, lichens usually grow on the bark of the trees or on the rocks. So where lichens are growing means wherever there are large trees, on the bark of the trees you can see lichens. If there are rocks, on the rocks, surface of the rocks, there also lichens grow. Either it may be tropical or temperate conditions, in both we find lichens. But lichens are indicators of pollution means, suppose in that area, if there are lichens, it is a non-polluted area. So lichens grow only in non-polluted areas. So lichens are the indicators of pollution. Suppose if you visit some area and you search for lichens growing on the rocks and bark of the trees, if there are no lichens at all, definitely this area is said to be polluted. So whatever may be the percentage of pollution, that is different. So if there is pollution, lichens will not grow. So that is why lichens are the indicators of pollution. So sometimes this is asked, lichens are the indicators of the pollution, give reason. Why? Lichens, they grow only in non-polluted areas and they do not grow in polluted areas. This is the indicator of pollution. So that is about lichens. So these are not included under any kingdom, virus, viroids and lichens.